Welcome to Chapter 9, where we're talking about cellular respiration, which is how animals and living organisms harness chemical energy. We know that living cells require energy from outside sources. On our screen here we see the giant panda, and some animals, such as the giant panda, obtain energy by eating plants. Some animals feed on other organisms that eat plants. We can trace all of our energy back to the sun. This image helps us visualize the processes of harnessing energy stemming from the sunlight. The light energy from the sun is absorbed by chlorophyll in the chloroplast. Photosynthesis then ultimately produces sugars, or organic molecules, as our screen shows, as well as a little bit of oxygen. In plants, the sugars then travel to the mitochondria for conversion into usable energy, or ATP. So how does the plant sugars get into our cells? We don't have chloroplasts, so we have to eat. Energy taken in by lower organisms in food chains harnesses the energy that initially was created by the sun. Now some of you may be thinking, well, we eat plants too. I mean, we have salads. Why do we need the middle organisms in the food chain? Well, if you think about how many calories we actually get from a salad, it's not many. It's like 30. Not much. This is because plants contain cellulose, and we're not able to digest that. However, some things that are lower in the food chain that we do eat, like cows, for example, they do. Cows are able to break down cellulose because they have a symbiotic relationship, or a partnership, with a special type of bacteria that resides in their guts. These bacteria can break down the cellulose, which provides cows with much more than the 30 calories that we would get. We would then eat the cow, and then we would get a lot of the energy that the cow got back to those plants. So this is all still stemming from the photosynthetic plants and the sun. Anyhow, back to our image. We can see, in addition to ATP and heat being released from the mitochondria, right down here, that CO2 and water are released as well. The CO2 in the water, once again, it's required back for photosynthesis. So these are kind of cyclical. So what we're going to be talking about involves a little bit of catabolic activity, or breaking down. So we break down things to get energy, to get fuel. And there's a couple different processes that we're going to be looking at as we move forward. So what we're going to be looking at, we're going to actually start by looking at aerobic respiration first. This would be cellular respiration. It occurs in mitochondria. And then we're also going to be looking at fermentation. There's a couple different types of fermentation. Fermentation, as you can see, does not use oxygen, so it is anaerobic. Whereas aerobic respiration, or cellular respiration, it uses oxygen, so it is aerobic. A with oxygen, and without oxygen. Cellular respiration, which occurs in the mitochondria of cells, involves both aerobic and anaerobic processes, depending on the step in the cycle we happen to be talking about with cellular respiration. Well, it should be mentioned that we'll be studying cellular respiration using glucose for simplicity. Other organic molecules follow a similar catabolic path. Other complex carbohydrates, fats, proteins, they all kind of go through the same process. The chemical equation that we see on our screen here is the simplified equation for cellular respiration, where we take a sugar, mix it with some oxygen, cause a little bit of reaction to occur to produce carbon dioxide, as well as water, and the energy that we are breaking all this down in order to get. That is the main idea for this whole process, is to get this energy, the ATP. Now, in order to actually make that ATP, we need to go through a process called redox reactions, which involves both oxidation and reduction. These two work exactly hand in hand, and you'll see how that occurs in just a moment here. What we need to know about redox reactions, it's when we're passing or accepting electrons. So let's take a look at how that happens. Right here we're looking at both oxidation and reduction, which we combine to call redox reactions. There's going to be a lot of vocabulary that goes into this that can get a little confusing. So what we need to understand is this. Things that lose electrons are oxidized by an oxidizing agent. And things that gain electrons are reduced by a reducing agent. We can see sodium here, Na, is losing an electron to chlorine. The electron is being lost, and chlorine is picking up an electron. How do we know that sodium is losing the electron based on these two images? Well, sodium is element number 11. That means it has 11 electrons and 11 protons. 11 negative, 11 positive equals 0, so neutral charge. But over here we see a positive charge. That means it has one more positively charged proton compared to the negatively charged electron. So it would now be 10 electrons but 11 protons, giving this a net charge of positive 1. So it's now an ion. Anyhow, sodium is being oxidized by chlorine, making sodium the substance being oxidized and chlorine being the reducing agent to that. At the same time, chlorine is being reduced by an oxidizing agent, sodium. Some redox reactions do not transfer electrons, but they change the electron sharing in covalent bonds. An example is a reaction between methane and oxygen. In this sample, we'll see the oxygen picking up the hydrogens from the methane. See, the hydrogens are now over here. Meaning that oxygen is acquiring the hydrogen electrons. So why not say that the carbon is being reduced from one molecule of oxygen then? This is because the difference in electronegativity between the oxygen atom and the carbon atom. The oxygen has 8 protons, or 8 positively charged particles, whereas carbon only has 6. Meaning that oxygen has a stronger pull on the negative electrons. Remember that opposites attract, and the electrons will be pulled to the stronger force. In the game of shifting electrons, in this case the hydrogen, electrons become weaker when they are bound to more electronegative compounds, meaning that it becomes increasingly more difficult to remove the electrons once they're attached to the hydrogens. So oxidation, reduction, oxidizing agent, reducing agent, redox reactions. Understanding what each actually is, I don't think is too bad, but you definitely will probably want to put some thought and time into getting them sorted out so you can remember, okay, this is being oxidized, meaning that this is a reducing agent, or vice versa. That's really the tricky part, I think. So put some time into that as you're going through your materials. So moving forward, we're going to be talking about some oxidation. And in this case, to get our energy, we have to oxidize some glucose. And we do that by using oxygen as a reducing agent. Now we can see the equation for cellular respiration. We can see the glucose and the oxygen. We can see that glucose is becoming oxidized, or it's losing its hydrogen electrons. Oxygen is being reduced. It is gaining those hydrogen electrons, and we also get some energy. In cellular respiration, which we can see on the screen here, multiple steps are required to gradually remove these hydrogens and get them to the oxygen. This really is the only way we can truly harvest the energy from the transfer of these electrons, which is where we get our energy, is by transferring electrons. The book compared this process to a gasoline explosion, where all of the energy would be instantly released without the ability to use any of it. By using a gradual shift, our cells can break down glucose and other substances gradually. We do this gradually to make the most use of the energy. So we do take these hydrogens and slowly move them over to the water. It's a slow process. This allows us to get the most energy that we can. Once again, straight explosions, everything is lost. So this is done by using enzymes. We use enzymes to make this happen. And during this process, hydrogen and its electrons are stripped slowly, once again, so they are not directly being passed off to the oxygen. We'll use NAD plus or NAD to do this. NAD is a coenzyme that acts as an electron carrier for hydrogens. Make sure you remember NAD plus or NAD is an electron carrier. We'll be looking at another electron carrier a little bit later. It's very similar to NAD. It's called FAD, F-A-D plus. So looking on our screen here, we can see NAD, right here NAD, picking up a hydrogen electron from right here to become NADH. NAD can deliver this electron that's attached to this hydrogen and then revert back to the NAD plus state. We briefly looked at coenzymes in Chapter 8 where they were described as essentially being a helper, and that's exactly what we're going to see going on with NAD with its reduction and oxidation cycle. This is done through the enzymes that NAD helps, which is dehydrogenase. Dehydrogenase removes a pair of hydrogen atoms, a pair, which includes, along with it, 2 electrons and 2 protons, since it's element 1. From the substrate that dehydrogenase acts on, glucose in this case, glucose becomes oxidized. The coenzyme NAD plus or NAD is then reduced. It's reduced because it's accepting the 2 electrons along with one of the protons, which is exactly what this image shows. And we see that because both of these hydrogens here are neutral, 1 proton, 1 electron. There's no charge, positive or negative here. But NAD does have that positive charge, meaning it's 1 electron short of being neutral. And over here we can see that, well, now it is neutral. Well, if it picks up a hydrogen atom that has 1 electron, 1 proton, it still would have a positive after it. So it picks up the remaining electron from the second hydrogen, making this hydrogen over here now an ion. There is no electron. This is simply a hydrogen proton, and that's all that makes it up. Anyhow, that's why this becomes a neutral, is because now it has picked up 1 hydrogen as well as 2 electrons, 1 from this hydrogen and the second from this one. This hydrogen ion will be removed. It's released as the hydrogen ion into the surrounding solution. And these hydrogen ions come into play pretty significantly later on. Here we see a picturesque format of what we were just talking about, the reduction of NAD into NADH. On the top right here we see that we have the 2 electrons and the 2 hydrogens moving along this way. One of those hydrogens will be attached to NAD, the other one will be released. And we can also see that the positive right here on the nitrogen is now neutralized because we have picked up the 2 electrons that we just kind of spoke about. So what is the purpose of NAD picking up these electrons, becoming NADH? The purpose is to carry those electrons. Recall that we had mentioned that NAD is an electron carrier. It is carrying these electrons, and it's going to take these electrons to the electron transport chain, part of cellular respiration, and very, very vital in the production of ATP. We'll be looking at that in the next video or so. So to kind of introduce you a little bit to the purpose of the electron transport chain, is to prevent this. Is to prevent that explosion. Unlike an uncontrolled reaction, the electron transport chain, which occurs in the inner membrane of the mitochondria, passes electrons in a series of steps. You can see a series of steps here, instead of one big explosive energy releasing reaction. Oxygen, you can see oxygen over here, will pull electrons. It has a strong electronegative core that pulls on the electrons in this energy yielding tumble. We can see that energy is slowly being released and it's slowly forming ATP, which ultimately will result in the formation of water when the electrons get done passing through the electron transport chain, they will join up with the oxygen that's pulling on them to form water, which will then be released.